Ever since people attempted serious explanation of the extraordinarily large bones found within the earth, artists have grabbed their pens, pencils, paint, and ink to reconstruct the often boring appearance of stony mineral bones, which many disregarded as mere rocks. From the 1700s to today, paleoartists have been a rather important section of the earth sciences. In my opinion, a broader definition would place paleoartists alongside many other nature artists, wildlife painters, prehistoric reconstructors, and landscape artists. That being said, I do think reconstructing an extinct organism is a bit more difficult than reconstructing already living animals, or their vibrant environments which you can easily go outside and observe yourself. In producing paleoart, the artist must be familiar with artistic techniques as well as general anatomy of living things in order to reconstruct anatomy of the extinct. The artist must also be aware of the colors, patterns, and behaviors of the living in order to construct something plausible for the dead. Obviously, that is all speculative. It's wrong. The math behind someone happening to get color, pattern, soft tissue, or behavior correct in the reconstruction of an extinct creature is beyond human comprehension. Therefore, making paleoart is attempting to be wrong in a real, plausible manner. That is, being wrong, but only so wrong, and of course, being aesthetically pleasing at the same time. Maija Kerala, or Erwin Tala as she goes by on DeviantArt, is a biologist, freelance science writer, and artist who reconstructs both living and extinct. She therefore has a certain level of credibility I want to make clear. She created a handy guide on how to plausibly reconstruct the colors of dinosaurs, and I found it most interesting. Therefore, what follows was taken in part from her articles and then twisted into something more long-form and containing more examples and in-depth discussion of the who, what, where, when, and why. Beyond my childhood's wildest dreams, we now live in an age in which we can rigorously observe, measure, and plausibly reconstruct general colors of extinct organisms. When a fossil specimen happened to die and become buried in a very special set of circumstances, soft tissues were preserved. Not just the mineralized skeletons, which give us only a general idea of what the animal looked like in life. Some of these super well-preserved fossils also happen to preserve pigments in the form of melanosomes and the cells which hold them, melanophores. Animals utilize many different strategies to produce the colors we see in their bodies. Pigments are the colored substances, but they aren't really colored the color we see. They contain elements and minerals which reflect only a very specific level of light and absorb all others. The brown in your dog's fur is not because it is actually brown, but because it is reflecting the brown light back into your eye. Our vision is very limited compared to many other animals. Some, like birds and many insects, see in the ultraviolet range of light, as well as the colored light spectrum, while snakes can sense infrared light. In reconstructing dinosaur color, we should be aware of this, as many dinosaurs probably could have seen into the ultraviolet spectrum like their modern descendants. Pigments are produced in specialized cells called chromatophores. Yes, those chromatophores. Chromatophore is the term used to describe those pigment-producing and holding cells found within cold-blooded animals. Fish, reptiles, crustaceans, and cephalopods. Their chromatophores are structurally different and are triggered differently than warm-blooded animals. The pigment-holding and producing cells in warm-blooded animals, birds, and mammals are called chromatocytes. Since the only pigment cell type found in birds and mammals holds melanin, the chromatocytes in these animals are called melanocytes. Melanins, the building block of color. As warm-blooded animals adapted from cold-blooded ones, they genetically retained these chromatophores. Over time, chromatophores were preserved by natural selection to negate some of the extreme color-changing abilities seen in many cold-blooded ocean critters we associate with changing colors. Many reptiles retain the ability to this day. When pigments within chromatophores are sequestered in a single spot, they make the animal appear paler. When they are scattered, the animal becomes darker. Reptiles, amphibians, fish, and cephalopods absolutely need this ability for camouflage and communication. Birds and mammals probably lost this for many reasons, but one big obvious one is their integument. Birds and mammals have fur and feathers covering their skin, which is where the color-changing chromatophores would be. If you have stuff covering the part of your body that changes colors, you no longer need color-changing cells. Instead of color-changing skin, birds, mammals, and some reptiles have opted for integument, which holds color, in order to camouflage and communicate. Melanins are pigments found in birds and mammals. Crocodilians, being far removed from whatever we want to call reptiles, are different from purely cold-blooded snakes, lizards, and turtles. Therefore, they too generally lack color-changing abilities. Melanins are found in grain-like organelles within melanosomes. Melanosomes are made by the melanocytes. These types of cells are further broken down into eumelanins and pheomelanins. Eumelanins result in blacks and browns, while pheomelanins result in muted reddish colors. Eumelanins, pheomelanins, and the lack of melanins can be used together to create different colors and patterns. Let's take a real quick look at crocodiles. True crocodiles of the Crocodylus genus and the related thin-snouted gharials are the only crocodilians capable of limited color change. That is, they can marginally change the hue of their colors, nothing like squid or chameleons. In a 2018 study, Dr. Mark Merchant, Dr. Amber Hale, and colleagues proved that crocodiles change color based on their environment. Those placed in white tanks eventually changed to be lighter in color, and those placed in dark tanks became darker. This is a similar kind of environmentally triggered color change we see in anole lizards and even chameleons. Chameleons are different, however, in their ability to force their colors to change via hormones and cell signaling as a result of a change of mood or health. On the other side of the crocodilian spectrum, alligators, caimans, dwarf crocodiles, and slender-snouted crocodiles completely lost this limited color change ability. These cold-blooded animals, considered reptiles by many, are lacking in all other types of chromatophores found in other cold-blooded animals, like fish, cephalopods, and crustaceans. These other types of chromatophores include the yellow xanthophores, red erythrophores, reflective iridophores, white leucophores, and blue cyanophores. Why would crocodilians, a cold-blooded, low-metabolic group, lose their chromatophore color-changing ability when they don't have fur or feathers to cover their skin? Researchers have presented some hypotheses. The one that makes the most sense to me, as someone who hasn't done their own research specifically on this topic, is that crocodilians had an ancestor which was fully warm-blooded with a high metabolism and a covering of feathers or feather-like filaments to cover their skin. This would mean that as crocodilians and their kin diverged from the archosaur tree, they developed a slower metabolism for a lifestyle more akin to that of the temnospondyl amphibians and freshwater fish which patrolled the waterways before them. In diverging in this way, they didn't have color-changing chromatophores in their genome to begin with. Those two small groups alive today which do change color would have had to evolve this trait independently. The physiology and genetics of crocodilians tends to support this hypothesis as they have a four-chambered heart, as seen only in the warm-blooded birds and mammals. The genome of the crocodilians also shares a lot of similarities with birds, and they have a unidirectional breathing system similar to what is seen only in birds. Another hypothesis posits that crocodilians may have undergone a nocturnal bottleneck at some point in their evolution. A nocturnal bottleneck is an evolutionary hypothesis used to explain several mammalian traits. It suggests placental mammals were nocturnal for most of their evolutionary history, from the start of the Triassic period to the K-pig extinction 66 million years ago. This amount of time spent as nocturnal animals left an effect on the anatomy of placental mammals, and those daytime mammals alive today are derived from these kinds of skittering nighttime creepy crawlies. Many mammals today are still nocturnal. Maybe this kind of evolutionary event occurred with crocodilians. They do share some similarities with mammals in how they developed their anatomies over time. Under this hypothesis, coloration would not have been as important to the small nocturnal archosaur ancestors and therefore lost their color-changing abilities in exchange for melanins. This hypothesis can be further supported by the limited color vision in modern crocodilians. Very good color vision is seen in birds and assumed in non-avian dinosaurs, so at some point a nocturnal common ancestor taking advantage of diurnal niches after the Permian mass extinction killed off the major diurnal animals makes a lot of sense. More research would need to be done to prove this further, if it can be solidly proven, that is. Melanocytes are very different in birds than to other groups which have them. They don't retain the pigments they produce. Instead, the melanocytes push their pigments into feathers, scales, and skin cells. Melanins usually form intricate patterns in feathers. A great example of this is in galliform birds. The jungle fowl, from which we artificially selected our chickens, show a subtle but striking pattern when all of their simpler patterns are seen together. The extreme, almost celestial spots and dots pattern of the helmeted guinea fowl, or the simultaneously orderly and chaotic rug-like patterning of the tragopan are some badass examples of what I mean by intricate patterns. These simple hair-like feathers seen in non-avian dinosaurs like Scuromimus, Utaranus, or Shibuya could probably be reconstructed with such intricate patterns, like what you see in porcupine spines or the hair of many types of mammals. Don't be afraid to splash some amazing contrasting browns, blacks, reds, and tans for forest dwellers, desert runners, or plains skippers of small to medium size. That's easy to say for theropod dinosaurs. They already look like birds and are the lineage which produced birds. But what about the other, not-so-birdy dinosaurs, the ornithischians? An exceptionally well-preserved specimen of Psittacosaurus may hold the answer. This parrot-faced bipedal ancestral representative of the ceratopsians is so well preserved, pigments can be observed under high magnification. These pigments were embedded in the scales of the body, as they only had feather-like filaments on the top of their tails. The feather-like filaments, dubbed bristles, are considered by most scientists to have a common origin with bird and non-avian theropod feathers. This means both avian feathers and the bristles of non-theropod dinosaurs originate from a common ancestor, further adding evidence to the idea that filamentous body coverings are a basic, ancient feature to the entirety of the Archosaur family tree. The Psittacosaurus specimen proves bird-like color physiology was present in most, if not all, lineages of dinosaurs in at least one point in their evolutionary history. The melanin found within the preserved melanosomes of the Psittacosaurus specimen suggests darker hues on the top 2/3 of the animal's body, and lighter colors the rest of the way down. The study on this specimen also found darkly pigmented scales in circular shapes bespeckling the shoulder area, a dark mask on the snout, a gradient from dark to light on the patagium that connected the limbs to the tail, and a darkly pigmented cloacal area. Needless to say, Psittacosaurus could not change colors like lizards. It has become clear that naked or scaly portions of a dinosaur pelt would likely have had similar color palettes to naked or scaly parts of modern dinosaurs, which can encompass just about any color or pigment used by modern dinosaurs: pink, purple, blue, green, yellow, orange, tan, brown, black, you name it. How do we know what color the preserved melanosomes represent when the animal was alive? When you observe the fossil specimen in question under an electron microscope, the cells which held the melanin are visible. Each type of pigment-holding cell has a different shape and density. If you observe the average shape and density of the pigment seen in the specimen and compare it to the shape and density of pigments in modern animals, you can estimate what type of color the fossil pigment corresponds to. I just tiptoed around any sort of explicit language. That's because, though scientists can get a general idea as to what colors these fossil pigments represent, an exact hue of said color is far blurrier. By blind testing fossil pigments with feathers of living birds, the reliability of this method increases. It's estimated to be about 82%, far from certain, but much better than nothing. As it stands, eumelanin, which is blacks and browns, are sausage-shaped, while paleomelanins, the reddish colors, are more like slightly flattened burger patties or discs. Many, if not most, dinosaur specimens with preserved pigments contain mostly melanosomes. Some also preserve semblances of structural color pigments, which I'll get into in the next episode, but it's a minority. Anchiornis was one of the first full-bodied specimens to undergo a pigmentation study. Before it was a few studies outlining techniques necessary to find and measure melanosomes in fossil animals. The 2010 study on Anchiornis found it had a mostly dark grayish body with white pennaceous feathers on the tail, legs, and arms, ending in black spangles. They also found the head's feather crown was a rufous or reddish color. Reddish speckles were also present on the face. Like I said before, these kinds of patterns are around 80% right on. So in life, this specimen may have had different hues to those colors, and some of the patterns may have been very slightly different. This is the best shot we got to reconstruct its colors for now though. Sinosauropteryx was a small coelurosaurian theropod from northeastern China. It's a catalyst when it comes to our knowledge of dinosaurs. It was discovered in 1996 and was the first official non-avian dinosaur described with feathers preserved. Before 96, many scientists were already in acceptance of the bird-to-dinosaur connection, and the sentiment that it was only a matter of time before an official feathered dinosaur would be found was commonplace outside of dogmatic old fogies. Sinosauropteryx opened the floodgates on dinosaur integument, but it would do so again when the electron microscope was placed over its remains in 2010. That electron microscope found the animal's integument to have a counter-shaded ginger top and cream bottom. Melanin-less stripes started at the base of the tail and continued to its end. The ginger melanin covered much of the legs and arms, and a ginger bandit mask covered the critter's face. Many smaller, exceptionally preserved theropods would get their colors as time progressed. The same 2010 study analyzing Sinosauropteryx took a look at a few other specimens from the same rock units, namely the clawed bird Confuciusornis, an isolated feather, and the dromaeosaur Sinornithosaurus. Samples were taken from these specimens and observed under a scanning electron microscope. The isolated pennaceous feather was found to have lost its pigment but retained the hollow voids where they once were. This still gave researchers the general shape to identify the pigments that were present as eumelanosomes, a gray, dark gray, or black. The Confuciusornis specimen has sausage-shaped eumelanin molds which held eumelanin and alternating reddish paleomelanins preserved. This means at least some parts of its body alternated between black or dark gray and rust. It's been likened to the zebrafinch of today, but such a complex pattern is unknown from the fossil specimen. A scanning electron microscope analysis of the dromaeosaur Sinornithosaurus presents paleomelanins and paleomelanin molds, as well as some amounts of eumelanin. This would suggest an overall rusty color for the tree dweller, but an exact color and pattern were deliberately left out of this paper as it would be too assumptive given the evidence. The earliest dinosaur specimens to spell out the connection between dinosaurs and birds is Archaeopteryx. In 2011, then grad student Ryan Carney and company analyzed the feathers of an Archaeopteryx specimen to attempt reconstruction of its colors. Their microscopes determined the presence of eumelanins in the feathers and concluded they may have been present throughout the flight feathers, as the feather they analyzed was a covert. which partly covers the primary feathers. This is consistent with the color seen in modern flighted dinosaurs, as the black eumelanins retain structural properties which strengthen flight feathers. Another study done on Archaeopteryx in 2013 found presence of both light and dark pigments and concluded the strengthening dark pigments were restricted to the ends of the feathers. The primary purpose of this arrangement is unknown beyond structural integrity. The same deposits in which Sinosauropteryx, Sinornithosaurus, and Confuciusornis come from also held the remains of a small therizinosaur, Bapialosaurus. The specimen preserves a nearly complete integument of short primitive downy feathers, akin to the dino fuzz of Sinosauropteryx. It also had a second covering of long, flat, stiff, hollow, bristle-like feathers. A 2014 study sampled feathers from the neck and found them to be roughly brownish. Preserved melanosomes aren't limited to Mesozoic fauna. The 5-foot, 1.5 meter tall ancient penguin Inkayaku preserves melanins within its short body feathers. The long, narrow melanosomes found within the feathers suggest presence of eumelanins, perhaps corresponding to gray, and pheomelanins corresponding to reds patterning the body. Paleoartists have reconstructed it with a dark gray top and reddish bottom. The melanins of Inkayaku were a little different than modern penguins in being much wider. There's a greater number of these melanins per cell in living penguins, with their shape producing dark blacks and browns. Another important example from a completely different lineage of non-avian dinosaurs is Boreolopelta. Described in 2017, this nodosaurid preserved much of the soft tissue of the top half of the body. Like many Diplocaulus bodies which bloated, floated, popped, and sunk, the body of the Boreolopelta specimen likely did the same. It floated its way out to a larger body of water before popping and sinking to the silt-covered bottom of a near-shore environment. Thanks to rapid burial, there's no evidence of posthumous desiccation of the corpse, which usually happens in other dinosaur mummies. It is this quick preservation of the top of the specimen which allowed researchers to use a mass spectrometer on the preserved keratin covering the scales and osteoderms to determine a general color. This thyreophore preserves a patterning of countershading, wherein the body is divided into a darker top color and lighter bottom color. I will go more in depth into countershading in a later video. There are a few ways to determine whether your fossil specimen retains melanosomes and what kind it retains. For Boreolopelta, UV light was used to identify hardened keratinous tissues on the body. In this case, those were the sheaths over the osteoderms. The more pigment present in the original tissues, the more those chemicals mask UV fluorescence in the fossil. This is why darker top colors were inferred. The sheaths covering the large shoulder spikes were the most fluorescent, suggesting close to a much lower concentration of melanin, corresponding to a light tan, cream, or whitish color. These methods were unlike the direct methods involved in the coloration of Anchiornis. However, the methods used for Boreolopelta were still rigorous. The preserved soft tissue film covering the top and bottom of the specimen retained different levels of preservation, often indicative of a change in melanin preservation. This essentially means the top is preserved more like a fossil with higher concentrations of melanin and the bottom with less. Another reason this is interpreted for this specimen is the distribution of keratin and collagen, which suggests a countershading dark top, light bottom. They did not actually find preserved melanin within the fossil, rather are inferring a reddish pheomelanin color due to chemical signatures which usually correspond to that color. Boreolopelta and Psittacosaurus are perfect to show paleoartists' complexity of colors and patterns that may have graced all dinosaur lineages. Boreolopelta is estimated to have been a couple-ton animal, so apply similar levels of adaptive coloration to your large, non-theropod dinosaurs. As much fun as it's been, we need to get to the other types of pigments found in modern birds in order to understand the full breadth of color, patterns, and structures we can reconstruct our dinosaurs with. Next time, I will go over structural color, the amazing reflective iridescence used for courtship by hummingbirds and peacocks. Stay tuned. Let me know in the comments below what dinosaurs you want to reconstruct and with what patterns of melanin. For more interesting stories about nature, the history of life, or what goes bump in the night, subscribe, like this video, drop a comment in the comment section below, and hit the bell icon to stay in the know with everything Edge. Thanks for watching. Special thanks to Elephant Tier patrons Abby Smith, Arda Bayer, Biotaverse, Cherry Shaw, Chris Frampton, Christoph Hubbinger, Dinosaur, Ed Peretz, Isaiah Garza, Jax the Hacks, Natty Cat, PA Brew News, Ray, Rudy Redgrave, Smiling Walrus, Staniforth Hopkins, Steve Bradshaw, Thea Svensson, and Extraterrestrial, as well as my Tapas tier Tyrannosaurus patrons, Admin, Antron, Aphid Kirby, Cyber, Dana Manchester, Danny Van Heck, Henry Brennan, Iberospinus, Iron Bladesman, Joshua Mana, Panic, Radio 404, Robert Kessler, Ruben Zachariah, Swaffles is Weird, Teeny Dragator, and The Dogman.